All right, and that job is to produce the myelin. Myelin is a fatty material, a fatty plus protein material that actually appears white. So all the myelinated areas in the nervous system are going to actually appear white, and you'll get to see this in the pig when we go back to dissection, and we're going to let you just kind of really get into and get him taken apart, him or her. Okay, multiple sclerosis is a problem with oligodendrocytes. And this is something that maybe in your reading you added into your notes somewhere, but multiple sclerosis attacks the myelin sheath in the central nervous system, and it kind of hardens it. And so now you're not making the connection between neurons, okay? It's what we call an autoimmune disease. The immune system attacks our own cells for reasons that we don't know. It's usually triggered by something like a viral infection or something like that got it started, but we don't know for sure. So that's a pretty serious thing, and people have various forms of that, or I guess degrees of multiple sclerosis, but it can be very debilitating where they can have complete paralysis, and sometimes it starts with something like slurred speech, pretty serious disease. There are some treatments for it, but there is no cure. Maybe you know somebody with MS. This is going to work for me. There we go. With the Schwann cells out in the peripheral nervous system, there can be issues with that too. Did we discuss these? I just want to make sure. No? Not sure? Why don't we just talk about them again? These are the two types of supporting cells out in the peripheral nervous system. The satellite cells kind of wrap themselves around the cell bodies or the soma out there, outside of your brain or your spinal cord. And then the Schwann cells have the same job again as the oligodendrocytes. They form a little differently though. They form that myelin sheath through different processes. The oligodendrocytes can branch out and attach to as many as 60 different neurons at the same time, so they're not wrapping their entire cell around the way that the Schwann cells do. Each one of these blue things would represent a separate Schwann cell, okay, that has wrapped itself around the axon of this neuron out in the peripheral nervous system, okay? What is the job in either central or peripheral nervous system of the myelin? What is it? Insulation of your axon. So what does that have to do with its function? Faster, the combination of the two, right. It's getting the transmission of that signal along the axon at a much faster rate, because you can skip over any place that there's myelin. You can't have that change in voltage. So what happens is you skip over these, and so now the signal is only in these nodes, or these nodes of Ranvier, so now you can get a faster transmission of the signal. So it would take a whole lot longer, especially with those really long axons that we have, like in our legs and our arms. It would take a lot longer to get the message there towards, your target cell, which would be here, right?
So the ones that are completely covered in our brain, maybe the fastest response there?
Well, they're going to be a lot shorter in the brain because you don't have as much area to cover. Let me go backwards here. The oligodendrocytes, there would still be open spaces. I don't think we necessarily need more coverage, because it's actually a shorter distance to go. It's just two different types of cells forming them. I don't know if I'm really answering your question, but
So like the purpose of these ones is more for installation and the other ones is more for faster transmission?
well, you still get the faster transmission here because they both do the same thing. No signal can be sent where this coverage is, so it gets the signal to skip over. I don't know if this is really, like, I wouldn't be saying that that's drawn to the same scale as the other one. Okay? So imagine these are different magnifications, in other words. So these aren't necessarily a lot larger. They just showed it, I think, so that you could see the detail and how they branch out into different places. So here's like your body of your oligodendrocyte, and these would be different. So you have more than one nerve fiber or axon involved from different neurons. Okay. So MS was a problem in the central nervous system, a similar disease in the peripheral nervous system affecting the myelin sheaths in your peripheral nervous system is diphtheria. And you probably were all vaccinated against diphtheria, DPT vaccine that you get 3 shots of, I believe, as a baby, probably, I don't know, 4, 6, and 18 months. I don't remember the timetable. It's been too long since I've had little kids. But so diphtheria is pretty much wiped out, but because it's a fatal disease. So it affects the myelin sheath in the peripheral nervous system, causing paralysis and eventually death. So it's pretty serious stuff. It's caused by a bacterium rather than an autoimmune. So it's something that we were able to control with vaccines. And so it destroys the myelin. Yeah, it says it can ultimately produce a fatal paralysis, so pretty serious stuff. Okay, the satellite cells we talked about, they're just surrounding there to support the soma of the neurons. Okay, so then if we look at the neurons themselves, these are the ones we call the nerve cells. Be careful of the term nerve because we keep using it and applying it to different things here. We're going to see that today. So it's better to use the term neuron and then supporting cell for the neuroglia, and then we'll be looking at axons. The processes should be specified as well. All right, the major regions, you have the body, which is also called the soma. Of course, that's going to be where the nucleus is, and these are very active cells, a lot of stuff going on in there. We'll take a look at the picture. And then you have the processes. Anything that sticks out from this is a process. Those things that receive are going to be the dendrites, and those that send the signal away are the axons. All right, in the body, there are a bunch of structures which you know as rough endoplasmic reticulum, or rough ER. This gentleman, Dr. Nissel, was the first to see those microscopically, so they get named for him. What do we call that when we use somebody's name attached with a structure? An eponym, right, an eponym. So here's a place where they've kept that. They're trying to get away from eponyms and naming things, so this is basically rough ER, okay? What is rough ER going to do in the cell? Do you remember? It has a lot to do with proteins being, first they're made on the ribosomes, right, and then they get dropped into the rough ER to get further processed. So there's a lot of protein synthesis going on here. Of proteins that are going to be used within the neuron. Okay, it might get shipped down the axon to the very end, so that's axon terminals to be used, or it might be forming structure. There's a lot of different proteins being formed. The neurofibrils are an example of these proteins, the cytoskeleton molecules that are actually giving it its three-dimensional shape. So those are found there in the body as well, and of course the nucleus with the large nucleolus. Anyone remember what the nucleolus does? You didn't really cover the cell in here. But what is it? It'll be right there in the nucleus. But the nucleolus, its job is to produce ribosomes. So it actually assembles the ribosomes there, and then they leave the nucleus and go out to the cytoplasm. So again, protein synthesis. That's why you have a very large, prominent nucleolus. These are very active cells, the neurons. The one thing that they're missing that most other cells have are centrioles. No centrioles. Remember what centrioles do? We talked about this, I believe. That do what?
Oh, that they're the fibers that pull apart the unit.
Right, the sister chromatids during mitosis, right? They're the spindle fibers. The centrioles form the spindle fibers that come in and attach to your 2 copies of your DNA and pull each chromosome or chromatid. Once they separate, they're called chromosomes. All right, so you don't have any centrioles, which means you can't do any mitosis, right? They can't reproduce, right? So this is the reason that neurons are unable to divide and fix themselves. They get stuck in that G0 phase of the cell cycle, remember that? So they're very active, but they cannot divide, which is a problem because if there's damage to them, we don't fix them.
Does that provide any benefits that they aren't going through mitosis?
I don't know what that would be. Yeah, you would think that there must be a reason for this, but it would be a whole lot better if they did fix themselves, because then we wouldn't have paralyzing injuries.
Cause more mutations?
Possibly. Yeah, you'd think that there's got to be a reason for everything like this, so maybe. It's the same DNA in any other cell, though. So any cell could be subjected to mutation. I don't know. It's a good question. I don't know if anybody's really come up with a good answer for that. The cytoplasm of the body has a special name that I don't think this textbook used, and that is called the perikaryon. The cytoplasm of a neuron is the meaning around, karyon, Karyon. It just means around the nucleus. Karyon is a Greek word for nucleus. Okay, so this is the cytoplasm of a neuron, the perikaryon. Okay, just checking to see if I wanted to add anything else. So in our diagram, you can see all of these, there's so much rough ER that it actually stains, and you can see a granular type thing if you saw in a microscope, because typically rough ER is too small for us to see in a light microscope. There's so much of it, and it clumps together, so we call these Nissl bodies or Nissl substance. Lots of mitochondria because you need a lot of energy. There's the nucleus. You can barely make out a nucleolus in the center of that. It might be more visible in your notes. And then all kind of these neurofibrils giving us our three-dimensional shape. And they run the whole length of the axon and into the dendrites as well. So that it's not like a floppy kind of mush cell. You've got a lot of, not necessarily rigidity, but a lot of structure to these neurons, okay? The axon itself begins at an area off of the body called the axon hillock, which is kind of a raised up area, and then that's going to come down and form your axon. An axon might branch. If it does, it's called a collateral, and that gives it the ability for one neuron to communicate with a lot of different target cells. Okay, so you can have one neuron communicating with more than one cell, which we know that anyhow because of these axon terminals. Towards the end of your axon, where it starts to branch out, those branches are called telodendria. Telodendria. Telo mean towards the end, dendria, tree-like branching out, telodendria, what is it, what's singular for that? That's the plural form. I guess I didn't write it down, I'm not sure. Telodendrion, there it is. So it's like mitochondria. Okay. So there's a lot of those, and then there could be thousands of these axon terminals. They only drew 6 of them here. So you've got all of these telodendria branching out, and then each of those is going to branch further into your axon terminals, which are that little bulb-like thing that we looked at pretty closely back in the muscular system chapter. Of course, you can see the myelin sheath here, and the Schwann cells wrapping around. So this would have to be found where in the body if we see Schwann cells here? In the PNS, right, the peripheral nervous system. Okay. What else? Numerous dendrites receiving the information. So the arrows are showing the direction of the signal. Okay. Something else that you might want to add into your diagram here is a typical synapse, which you pretty much covered, or you should recognize from what we did with the neuromuscular junction. So a typical synapse, remember, so I'm looking at just one of these axon terminals. It's going to come down. So if this is our axon terminal, what do we find inside the axon terminal? A lot of vesicles, right? Hundreds of them. What's in these vesicles? Neurotransmitter. Now, there's a bunch of different types of neurotransmitters. They're all chemicals. Okay? And their job is to be released here when calcium comes in. We won't go through all this, just looking at structure mainly now. But so if calcium is allowed to enter into the axon terminal, it causes the vesicles to come in and fuse with the membrane. Should draw, they're like, and then we can release their contents. What else does a typical synapse have to have? There's 3 basic parts to any synapse. This is one of them. The cleft, okay, is the space in between. And so then what's the third part? Any target cell. It could be another neuron, it could be a muscle cell, it could be a gland, whatever, okay? So you need the membrane of your target cell. So this would be its membrane. There's the third part. Or, well, let's say that's the second. And then as Ali mentioned, this space in between is the synaptic cleft. Okay, so those 3 parts, you have your axon terminal, the space, the cleft, and then your target cell membrane. Those are the 3 parts of any synapse. So that's how you're going to end up sending the signal. The beginning axon terminal is called the presynaptic cell, and your target cell is the postsynaptic cell, right? You should recognize those terms from those interactive physiology things you did. So the end of the neuron that's sending the signal would be presynaptic. The one that's getting the signal is the postsynaptic. Okay. So I think that just basic synapse structure definitely needs to be embedded now in order to understand any of the other things that are going on here. Okay. All right, does everybody have it? Can I erase? All right, this is a great picture, I think. It's a scanning electron micrograph. So it gives you just surface things. So we can't see internally into the soma there, or the body, but you can see the dendrite. And this is, doesn't it show you where the axon hillock is going into the axon? So since it's calling this a dendrite, I'm guessing maybe that one, but, or this could even be one of those types of neurons that this textbook didn't even talk about, and that is called an anaxonic neuron, where they're actually, you can't tell what's a dendrite and what's an axon. It has it, but we don't really understand them very well, but okay. So looking at those processes then, we talked about dendrites. They're going to bring the message in. There can be hundreds of them. Axons send the signal away, and there's usually only one axon, and it comes from that, or starts at that axon hillock. So they show the picture again. Axon terminals, we talked about all of this. Okay, so we know what our synapse looks like. The myelin sheath. Okay, so let's take a little bit more closer look at what's happening with these Schwann cells in the peripheral nervous system. The Schwann cells will actually wrap themselves around the axon. So each section of the myelin sheath is one Schwann cell, and it squeezes out its cytoplasm so that the inner layers of this, it's kind of this jelly roll type of a thing, the inner layers are just plasma membrane. Remember what plasma membrane is made of? The phospholipid bilayer. So here's where we get this fatty myelin material. Because within those phospholipids are embedded what? Random proteins, right? Remember that fluid mosaic model where you have the fluidity is your phospholipids, and then you have these proteins kind of floating around in there that do a number of different things. So that's where we get the protein and the fat material. So we squeeze out cytoplasm, so now it's just plasma membrane, plasma membrane, plasma membrane, and then you end up with just your cytoplasm in this outermost layer, and that's what they're calling the neurolemma. The layer of the Schwann cell that still has cytoplasm in it is called the neurolemma, okay? And the other portions is just membrane, so that's what they're calling the myelin sheath. So it just keeps wrapping around like this. So again, every little section is a separate Schwann cell. So we could have, literally millions of Schwann cells in here myelinating the axons in our peripheral nervous system. Back up. Again, the oligodendrocytes wrap around, but they don't do that jelly roll kind of a wrap. They just wrap themselves around, and it's just that one single layer forming the myelin sheath. Okay, so that term neurolemma does not apply to your oligodendrocytes. All right. Neuron cell body location. So this is the bottom of page 5 now in your notes. The bodies, or the somas, are mainly found in the central nervous system, but there are some outside of there. Okay, so within your central nervous system, we have this term gray matter. You've probably heard that. So if it's gray in appearance, then there really isn't any myelin there, because remember, myelin is white. It's like a fat. So the gray matter would be cell bodies and the unmyelinated fibers. So all your dendrites, there are some axons that don't get myelinated, but okay. A cluster of cell bodies within the white matter is called a nucleus, or the cluster would be a nucleus. So nuclei. So again, why do they have to use these same terms for more than one thing? Okay, when you hear nucleus, you think of the nucleus of a cell. Now they're applying that term to a bunch of cell bodies, which to me doesn't make sense, but that's the term they used. So the term nucleus here in this form is referring to a bunch of the cell bodies within the central nervous system. Okay. And if they're in the white matter, then that's a place where there is myelination that has occurred. Okay. A similar term outside the central nervous system, so we're talking about peripheral here, now that those clusters of cell bodies are called ganglia. A ganglion is singular. So ganglia refers to peripheral nervous system, nuclei refers to central nervous system. So many different terms. All right, so there's just some terminology to get down. Here's some more. The term tract is for a bundle of nerve fibers. When they're talking about nerve fibers, they're looking at processes, the axons and the dendrites. So if a bunch of those together in the central nervous system is called a tract, and a bunch of those in the PNS is called a nerve. So again, this is where I was talking about before. When you see that term nerve, specifically it's supposed to refer to just the axons and dendrites within the PNS, but we use that term for a lot of different things. White matter, again, is where we have myelinated fibers, and that term is usually only referring to CNS, so that would be the tracts, gray matter, your unmyelinated fibers, and your cell bodies again. Okay. Neurons can also be classified based upon what their job is. So the sensory ones are going to be bringing in information from the sensory receptors. So your cutaneous sense organs, like in the skin and so on. So they're going to bring the information into the body or central nervous system, and then your motor or efferent neurons are going to send the signal away to your target cells. That might be muscles, different types of muscles, the organs, the glands. Okay. A specific type of these sensory receptors are called proprioceptors. Proprioceptors give us an idea of where the body is in relation to other structures of the body. So it's sensing positioning, especially within our muscles. Okay. So you can tell where your arm is, where your leg is, that kind of stuff. All right, they're detecting. A lot of them are found in tendons and ligaments, so they can tell if you're stretched or not, and what position the body's in. Okay. There's so much terminology to this chapter. More and more and more terms. Trying to keep everything straight is going to be tough. Okay. The sensory or afferent neurons, we have about 10 million or so of those. About 10 million. So I'm going to give you some numbers just so you can kind of get an idea compared to each other. Okay. There are two subdivisions there of sensory or afferent neurons. It would be the somatic sensory neurons and the visceral ones. The somatic sensory neurons are monitoring the outside world, so temperature, light touch. So think of things that your skin would pick up. Your somatic sensory neurons. And then our visceral sensory neurons are going to monitor like the internal viscera, organs and organ systems. For the motor or efferent neurons, there's only about 500,000 of these in the body. It's a big difference. Only about 500,000. Their cell bodies are always in the CNS. Your cell bodies are always in the CNS. Whereas with your sensory ones, the cell bodies were outside the CNS. And again, you have somatic motor neurons and you have visceral motor neurons. The somatic motor neurons, again, are taking information to the skeletal muscles. The visceral motor neurons, everything else. Your smooth, your cardiac, your glands. Okay, we need to repeat that. somatic motor neurons send the message to skeletal muscle, visceral motor neurons, everything else. Think about the autonomic, remember, in your somatic. Okay. And then the third type of neurons are the interneurons, which are sometimes called association neurons. Do we have any listed here? There we are. I'll go back to the pictures. These are going to be connecting your sensory and your motor neurons. That's what they're called, inter. They're in between the two. We have 20 billion of these. Just to again put these in perspective. 20 billion or so interneurons compared to only 10 million and a half a million of the other two types. Okay, so they're always going to be connecting your sensory and your motor. And they're going to be found in the central nervous system. So with the picture here that you should have labeled, the blue is always showing us our sensory, the red is the motor, and they're using green for the interneurons, or those association neurons. So here your spinal cord, remember, is just the bundle of nerves found within the spine. The cord, when you see cord, it's just the yellow portion here of the model. So remember how it's located and protected within these spinous and lateral processes of the vertebrae. Okay, and then you've got the branching processes, as you can see here. They use yellow again. All right, so this cord, if we did this cross section, if they had added more detail to this, we would see this butterfly-shaped appearance with the gray and the white matter. Okay. So within there, you find those interneurons, okay, and they're going to make the connection. So somatic, and notice the cell body is not found within the central nervous system with your somatic neurons. A bundle of cell bodies here there's that term ganglion. Okay, so that's what that's referring to. And then your motor neuron is coming back out. Okay, and sending the signal. So the direction would be coming in this way, process the material, decide what to do with it, send the message back out through your motor neuron. Afferent transmission, efferent transmission. I know we used Kayla's little example there to remember afferent and efferent. Another one that the book uses is the word same. The SA can go together, and the ME, sensory, afferent, motor, efferent. That might help you remember them. I'm not going to mention Kayla's again. All right, so if you take the word sensory, afferent, motor, efferent, just make sure you put the letters together. Okay. All right. I'm going to stop there. But I do want to remind you